Welcome back. So it's my absolute pleasure to introduce Dr. Ricardo Mouro Pinto. He's an assistant professor of neurology at Harvard Medical School. In addition to that, he's a Brody, who's a member of the MPG program. He also runs a lab at the Center for Genomic Medicine at MGB, where he is focused on neurodegenerative diseases, including Friedreich's ataxia and Huntington's disease. And he'll tell you more about his research today. Thanks, Ricardo.
Mike's on. Perfect. Thank you so much for the invitation to present our work here. Just a quick disclosure. So as Jillian mentioned, we're very much interested in repeat expansion diseases. And I'll touch a little bit on Huntington's disease, which is an autosomal dominant disorder caused by a CAG expansion in the HTT1, in the HTT gene in exon 1. And once you inherit an expansion beyond about 40 CAG repeats, you'll develop Huntington's at some point in your life. And the disease is primarily characterized by neuronal loss throughout the brain, but primarily in the striatum. As you can appreciate here, the atrophy in the brain. And interestingly, there's a strong relationship between the length of the repeat and the severity of the disease. So the longer the repeat, the earlier you'll develop symptoms, and you'll also have a faster and more severe progression. And the disease can manifest in a variety of different ways, primarily motor symptoms, but also cognitive and behavioral. And an interesting aspect about these repeats is that they're unstable. So common phenomenon in Huntington's disease is called anticipation, so meaning from generation to generation, patients tend to develop the disease earlier than their parents. And this is primarily because the repeat changes from generation to generation. And as you can see here, from maternal transmissions is sort of stable. So the child will have more or less the same repeat as the mother had. But if the transmission is from the father, there's a higher chance that it will expand and it will become much longer, and therefore that the child will end up developing the disease earlier. But something that hasn't been so well appreciated is also the fact that the repeat is also unstable in the brain. It's not just in the germline, but also somatically in the brain. And you're seeing here a brain from an individual that died not from HD. This is a brain about 13 years before the predicted onset. So there's no real signs of neurodegeneration in this brain, but you already see some neurons or some cells from what you can appreciate here that have expanded drastically beyond the two inherited alleles. So this would be the wild type, this would be the mutant allele, and you can see expansions all the way up to 1,000. And this starts in the striatum. This is a different brain, late stage, heavily degenerated, and it starts, you start seeing it throughout the brain. This instability is present in different parts of the brain, and the striatum, this particular brain, strikingly low, but the hypothesis is that at this point you hardly have any neurons left. Most of the striatum is gone, so you're not really seeing it. And I'll present a couple of data. Some of you're probably aware of some of Steve McCarroll's work, but there's been some focus on trying to understand exactly where this is occurring at the cell type specific matter. And two studies that came out in the last couple of years, one by the Nat Hines group in Rockefeller, where they did fan sorting. So they enriched for different different cell types from the brain and sized the CAG repeat in each of these enriched populations. And you can see that in medium spiny neurons, which are the neurons that are primarily affected in Huntington's disease, accumulate these repeats. And Steve validated that with an independent approach, with single cell level, sizing the neurons, characterizing the different cell types based on transcriptional information, but realizing that these large expansions are only present in medium spiny neurons. But interesting, what this allowed, this single cell resolution allowed Steve to ask a different question. How different are these neurons from each other, these medium spiny neurons? And turns out they're not that sick, they're not that different. Neurons that have these CAG repeats that are 40, that we would consider pathogenic, they're indistinguishable as they increase in size, and it's only once they cross a certain threshold of about 150 CAGs that become dysfunctional and then they die. So this is it's very important because it changes completely the way we understand the disease and how we approach it from a therapeutic point of view. And it makes a lot of sense also to intersect it with genome-wide association data, so data from patients all the way from the last 10 years, going up from 4,000 all the way to 16,000 patients. And this is not the traditional GWAS, this is not a case control, these are all patients, and we're just looking for the effect of the effect on age of onset. These would be modifier genes. And we're starting to see a pattern. Most of these are DNA repair genes. You do find Huntington gene, and I'll get into that later. This is an interruption in the gene, but most of the other genes that are coming up are DNA repair genes. And this was very interesting when we started looking at this, because this was actually something that we had already discovered in the mouse. So in genetic crosses with mice, we knew that some of these genes were essential for these somatic expansions. So when we cross an HD mouse with an MLH1 knockout or with an MLH3 knockout mouse, we were completely abolishing. So this is expansion. This is just a different way of quantifying the CAG expansions. This would be the inherited allele. This would be expansion. When you knock out these genes, you completely abolish the expansion. So you stabilize the repeat. And this led us to build a model at the time that was fairly incomplete. But with the poor understanding we had of that model and the increasing number of genes that were coming out of the GWAS, we wanted to try to establish a functional role. Most of them we didn't really know what function they had in Huntington's disease. So we designed a CRISPR screen, an in vivo CRISPR screen, where we cross our HD mice with a mouse that expresses Cas9 endogenously, and then we just have to deliver the guides of interest in an AAV HD animal so that we allow expansions to either occur or to stabilize them if we turn off a gene that is a driver of instability. And this is just a validation experiment. So we're seeing here that these different way of quantifying this. We can convert this into an index, the rate of instability. So we know that it is age dependent. It keeps getting larger and larger in the striatum. Actually, this is data in the liver. At this point, we leveraged the fact that AAV8 is very strong at transducing the liver, and the instability is also occurring in the liver. And we noticed that if we knocked out the genes that were essential for driving expansions, we completely stabilized the repeat all the way to the same level that when we started the experiment. Conversely, FAN1, which is a strong modifier that came out of the patient GWAS, we accelerate the rate of expansion. So here is showing the result of the screen. This is data coming from about 600 mice, total of 60 genes. And we probed a variety of different genes coming from the GWAS data set and a variety of different DNA repair pathways, and some of them coming from the literature. But so we probed these different 60 genes and ranked them based on effect on somatic expansions. And interestingly, what we're still finding is exactly the same. It's the mismatch repair pathway that is either stabilizing or accelerating the rate of expansion. And here, at this point, we're only doing it in the liver. It's a neurological disease. We collaborated with Ben Everman. We packaged this in PHP-EB to target the striatum, and we could validate these genes in the brain. Alternative ways of validating, we actually, for some of these, they were novel targets, like PMS1. We generated the mice and we crossed them. with our HD mice, and we observed that PMS1 is an essential gene. It's not as strong of an effect as some of the genes, but it's significantly reduced instability in the brain. And we collaborated with the Gazella lab to develop a cell model of expansions. So in culture, the repeat would expand in a short period of time. And when we knock out these modifier genes, we can either stabilize it over time or accelerate it. And we can use it to validate our de novo sort of hits. Still from the point of view of mechanism, exactly how this occurs, we're very interested in a gene, MLH3, because of its role as an endonuclease. So we wanted to know, because there was still a question about, does it only require these proteins to be recruited to these secondary structures, or do you need catalytic activity? So we've used a mouse model that had a point mutation in the endonuclease domain that still expresses, still forms a dimer, but question, does it still promote expansions? And the answer is no. If you have no endonuclease activity, you completely stabilize the repeat. So interesting, this also provides, and it's become clearer later, but it provides us a different way of how to target these genes. And this is, we just recently published this, but it's sort of our current understanding of how this occurs. So in red here, we have the CAG repeats during transcription, or during replication. In neurons, it will be during transcription. You'll unwind the DNA, and you'll have a propensity to form these slipped strand structures, these hairpin structures, that get primarily recognized by MSH2, MSH3, or mutase beta, and then recruit other components of the different mutal components of mismatch repair. We believe that MLH3 is required to cut the opposite strand, and then you'll have polymerase delta, which is also a gene that I didn't have time to go into, that came out of our screen, to synthesize a new copy of the DNA, and by doing so in both strands, you add additional repeats. And as I mentioned at the beginning, we're very much interested in repeat expansions. And it's not we primarily work on Huntington's disease and Friedreich's ataxia, but this is a group of as many as 60 different rare diseases. So here you've got the CAG, CTG repeats that tend to form hairpins, the Huntington's disease, spinocerebellar ataxia, myotonic dystrophy, and Fuchs corneal dystrophy. But then you have others as well that are a little bit longer. So something I didn't mention, they can occur in a variety of different locations, both coding or non-coding, just in regulatory regions. They can have a variety of different lengths. So CAG repeats in Huntington's disease, around 40 to 60 CAG repeats at the majority of patients, but other diseases are in the thousands that have repeats. Another interesting aspect is that depending on their sequence, they can form a variety of different secondary structures. We believe that that's what makes them likely to be recognized by the DNA repair machinery and expand. But because they all have somatic instability, we also want to leverage, can we identify a common genetic therapeutic target, target that would possibly be have therapeutic value not only to Huntington's disease, but a variety of other repeat expansion diseases. So we performed the same similar experiments, similar screens, and summarizing here just those results for Huntington's disease from Friedreich's ataxia, which is a GA repeat, and Fragile X. We're doing this now in spinocerebellar ataxia 1 as well, and we're trying to combine this data to try to understand in what directions these modify the instability. And we've identified a few. Want to caution that it's a tricky thing to target DNA repair. It's there for a reason. We think in this case it's actually misrepairing, But we need to be careful because some of these are actually quite strongly associated with cancer, particularly colon cancer. But there are 3 that are very minimal effect, or many, very little documented implications for cancer that primarily involve in spermatogenesis, in replication, in meiotic recombination. But so we've identified these 3 particular genes that we're kind of excited, and the field is also excited about. Few examples on how we can target these. I'll give you a quick one on how to use a small molecule, a splice modulator, something that was actually under testing for Huntington's disease, but with Huntington's disease as being the target, so just as a Huntington lowering strategy. One of its off targets is actually PMS1, one of the genes that we've shown as a modifier. And we can see that with this drug, we can reduce the level of PMS1, and we can also reduce the effect on instability. Here, just sort of validating the therapeutic effect of so transcriptional dysregulation and also accumulation of mutant protein. When you knock out PMS1, you reduce the accumulation of the mutant protein in the nucleus, in the striatum, and also you sort of rescue some of these transcriptional changes. So sort of validating this therapeutic target. Going back to MLH3, we're particularly excited about this because knowing the dependency on the endonuclease activity and the fact that this gene is expressed as 2 variants, one containing the endonuclease and one without through alternative splicing, collaborated with Ed Grabzik to use ASOs to sort of favor the splicing of this gene towards the endonuclease lacking version. And interestingly, this isoform is even expressed in some regions. You have the endonuclease lacking version expressed at higher levels than the one containing the endonuclease. And briefly, try to go through this quickly because we've tested this with ASOs and we could suppress somatic expansions with ASOs, but we couldn't use them so well preclinically. We couldn't get them to target the brain as effectively, even though we're able to reduce somatic expansions in the kidney. It wasn't as easy to do with an ASO, and based on our collaborations with David Liu and with Ben Kleinsteiber, we sort of pivoted with this strategy, still keeping the same target and the same mechanism, but with a different approach. And we hypothesized that given how conserved splice acceptors are across species, we hypothesized that if we simply disrupted this AG sequence, we would be able to alter the splicing of that region. So, as I mentioned, we collaborated with David, who's invented base editors, and we don't need much of an introduction here, and with Ben Kleinsteiber, who has engineered these enzymes so that they can recognize a variety of different PAM domains. So we can essentially target different regions in the genome and not necessarily limited to an NGG PAM. So how did we do this? We essentially tiled the entire region with a variety of different guides, with a variety of different ABEs and a variety of different CBEs. And fortunately, we identified a couple that actually seemed to be quite effective in HEX cells. And this is what we've been pursuing further with in further validation. Importantly, we've showed that the single point mutation is sufficient to completely change the splicing. So we no longer have any expression of the transcript that has the endonuclease. And in fact, when we test this in a cell line that has instability, so you can see here across a month of culture, the repeat expands. when we have this single point mutation, we completely stabilize it. Next, we've already started our preclinical work with HAB to generate the mouse model. So rather than testing this and using guides towards the mouse sequencing, we've engineered a mouse that contains the human sequence flanking it. Interestingly, and this is something that Cures Biology, just changing these 30 base pairs, all of a sudden the mouse is splicing like a human cell would. So all of a sudden mice normally just express variant 1, and all of a sudden with this humanized sequence, there's alternative splicing. Perform very quickly preclinical work with some, I would say, not optimal AAV preps. This is something that we still need to repeat. But we identified a couple of animals where we had decent editing in the striatum. We show that in the striatum we're altering the splicing, and more importantly, we're reducing the instability by 40%, even with these sort of poor AAV preps. So we're quite excited about this and moving it forward. Again, important considerations here is really in terms of safety and in terms of delivery. These are things that still need a lot of work. Won't touch too much here, but here just going another example of our collaborations here at the Broad and giving you a different strategy, also targeting expansions. We know that individuals with interruptions in the middle of the CAG repeat develop the disease later. So work done by Janetta here at the David's lab hypothesized that if we were to use base editors to introduce these silent CAA interruptions within the CAG repeat, we would slow down instability, and in fact we do. And so when we have about 30-40% editing in the striatum, we can reduce the rate of expansions and also promote the rate of contractions. So just a little bit of a cartoonish overview. We think that throughout the life of an individual, the CAG repeats keep expanding in a cell type specific manner until they hit a certain threshold where it starts becoming toxic and causing neurodegeneration. You start losing these neurons, and upon a certain time, that will manifest as symptoms. From the genome-wide association study, we hypothesize that there are certain genetic variants that will change the rate of that process, how fast the repeat expands or how slow it expands, and it will manifest as different ages of onset. And so we hypothesize that if we use a therapeutic to slow down or stop somatic expansions, you will either slow the progression, or if you go before onset and you're effective enough in stabilizing the repeats, that you can effectively cure these diseases. And so with that, I want to thank everyone in the lab, our collaborators, and the funding and Letters to Cures for support. Thank you.
As we mentioned, we'll do Q&A jointly. So our next speaker is Dr. Jen Pan. Jen is the Director of Translational Neurobiology here at the Broad Institute and the Stanley Center, where her lab focuses on high-throughput evaluation of ion channel physiology and next-generation therapeutics. Importantly, she's also an advisor to the CACNA1A Foundation, whose leadership is in the audience today. Welcome, Jen.
Hello. Thank you so much, Jillian, for that introduction. And I wanted to thank Anna and Jillian for organizing such a fantastic symposium. It has been inspiring to hear the messages, the approach, the research and stories. So today I'm going to talk to you about one of my favorite genes, CACNA1A, which encodes an ion channel. As an example of how we think about rare genetic coding changes with intermediate effect size is going to contribute to the disease burden, how we think about the treatment strategy. Okay, there we go. So I wanted to start with a few words about channelopathy. The monogenic Mendelian mutations that causing diseases in ion channels has been studied since the beginning of the molecular genetics. These are recurring mutations produce a common shared disease phenotype, and then there are many reports of such. There are very few mutations, but once it happens, it produces very repeatable, very common disease phenotype. And then in the past 2 decades, channelopathies in complex polygenic disorders has been discussed and has been discovered by the GWAS. On the right here is a figure that was published 15 years ago by Steve McCarra and Peter Fischel and many others. They demonstrated that at that time there might be missing heritability from GWAS studies. Are these rare and low frequency coding variations with intermediate effect size in the spectrum of the disease? And this is what I'm going to focus on today. So why do we care about these rare and intermediate frequency, the intermediate effect size, low and rare frequency, rare coding variants? And so fast forward 15 years to where we are now, our ability to detect them has increased exponentially. So here is a figure of how many SNA variants has been identified in the past 20 years. And you can see with the DNA sequencing, exome sequencing, we have started to see hundreds of the variants, but many of them with unknown function. So annotating a large number of coding variants are necessary to explore in a cohort, in a set disease cohort sense, has become accessible, and we have increasingly able to identify these low-frequency coding alleles. And channelopathy is a great place to start addressing the connection between genetics and functional consequences, because ion channels, they have these assays to precisely study their function by electrophysiology using patch clamp. So you can get very detailed look at the functional outcome, the voltage dependence of activation, or ligand binding, or kinetics of how they open, how they close. And then in addition to that, annotating using this class of proteins as an example, annotating a large number of them can be very powerful to further propel the human genetic study of complex diseases, as well as stratifying patient population. They're very critical to identify the patient to go after the treatment and design the precise treatment. So with that introduction, this is what I'm going to talk about today. These are, so here is the outline. So first I'll introduce what is a CACNA1A spectrum disorder. And then I'll describe to you a couple of studies addressing the missense changes of CACNA1A in a cohort of neurodevelopmental disorder. And then the results, what can we learn from this study in a cohort study of neurodevelopmental disorder? And our subsequent analysis trying to link the molecular phenotype with the clinical phenotype. And then lastly, for a few slides, I'll talk about the strategies towards a potential therapy and potential nodal mechanisms underlying these coding variants as powered by the letter to cure as a pilot. So first of all, what is CACNA1A? CACNA1A encodes a PQ type CAV. It's a voltage-gated calcium channel. It's a 2.1 voltage-gated calcium channels. And then there are 10 genes encoding these large transmembrane proteins. They respond to voltage changes and are expressed throughout the excitable cells in the body. Many of them are expressed in the brain, as you can imagine, because neurons communicate with electrical signals. So CACNA1A is right here. It's one of this is family, the CAP1 family, CAP2 family, and CAP3. So CACNA1A is right here. It's expressed widely throughout the central nervous system and also present in the peripheral nervous system. it mediates the neurotransmitter release at the majority of the presynaptic terminals in the central nervous system and mediate excitability, both neurotransmission and at the soma for the excitability. The reason it was originally called P-type channel is because it was initially identified in Purkinje cells. And then back then, that was initially called P-type channels. And then the structure of let me see if I can, it doesn't play. The structure of the CACNA1A has been published. And then so this could be a starting point for structural assisted rational design. And it's also one advantage when we're going after the precision medicine. So this is a table that I take from CACNA1A Foundation. There are many phenotypes or clinical manifestations that have been annotated for people with CACNA1A mutations. Some of them has been well described, for example, the hemiplegic, familial hemiplegic migraine. Some of them are sporadic, and sometimes you see them in the people harboring these mutations, sometimes you don't. And it's spread across quite a different domain. So it's very difficult to grasp in what is the connection between the mutation or what type of mutation to the clinical manifestation. So we started to ask the question utilizing a published result of the neurodevelopmental disorder. So what we did in this context is to take the exome changes in the CACNA1A from a 31,000 trial patients. Okay, so in this cohort, 49 individuals has contained missense changes, and only 4 individuals with protein truncating variants. So missense changes are dominating in this cohort. A few variants have been documented before, but the majority of them are with unknown function. Okay. So we take this entire allelic series of de novo mutations of the CACNA1A and summarized here, including the 42 unique de novo variants. And this is coming from 36 unique coding changes that I just described to you from the previous cohort. And we added 6 additional novel de novo mischange coming from the clinic in Dr. Helbig's in CHOP and then Dr. Pajuri at Boston Children's. This comprised of 42 unique de novo mutations. Taken together, we also added 8 most frequent missense changes in the general population, and we wanted to include the nomad mutation to give us a sense of what the true wild type range of that functional activity is. And then here is a topology of the channel where, for those of you who are not familiar with this large protein, they're roughly divided into 4 transmembrane domains. And each domain is composed of the 6 transmembrane segment, and then they come together, wrap together to form the central ionic pole in the middle. So here, the red dots represent the disease-associated missense changes across the channel. You can see most of them are located in the transmembrane region. And then the green dots are represented here, representing the nomad variants, and then they're mostly located actually in the loop region. And right here, you see some segregation of where the mutations coming from. And I'll skip this slide, and this is describing the overall, how do we choose this, the nomad variants. So as I mentioned, one of the advantage of this class of proteins is that you can study them with a huge amount of precision. So the way we study them is electrophysiology. These channels are broadly described in open state and then closed state and inactivated state. So typically they're sitting at closed state, and when the membrane potential change, they become open with open probability increasing. And then sometimes they go into inactivated stage where it's very difficult for them to open again, but then they have to go back to the closed state before they can open. So we can devise the protocols to precisely probe their transition between the open, closed, inactivated, as well as the kinetics of how they do that. And then here, I probably will show some graphs. This is what we call it, the IV relationship, current voltage relationship. And then most often we define how they open and how they inactivate with this graph we called voltage dependence of inactivation and activation. This is the activation curve, and this is the inactivation curve. And then the technology that enabled us to query almost 50 plus variants in this gene is a couple of technologies. One is a maxcytin that can transfect large cDNAs very efficiently, and another technology that allow us to be able to patch on a whole chip of 384 well plates. That is a huge improvement on the typically very laborious manual patch clamp. And then we did that for all of these 50 variants. I'm going to jump through the technical side, and then this is the first result. When we first saw this, we are very surprised. Basically, you can view this on the y-axis is how much of occurrence that we can see from each of the variant channel. And this is, I don't know why I don't have the label, but this is basically the wild type. And each of this bar represent one variant, and different color represent different domains. So, and then here at the gray bars are all nomad variants. So right here you can see nomads behave more or less like a wild type. However, near half of the variants from the disease, from this neurodevelopmental cohort, do not show current density. We're quite surprised by that because it wasn't obvious, because we thought if just losing half of the channel, losing half of the copy of the channel would be the main contributor. We should have seen that in the PTV truncating variant. And hold the thought, I'll come back to that. And here is another example, just give you some feels of how these variants behave. And so these are three different examples of the variant that calls the gain-out function, what we call, in the sense where they all shift the voltage dependence of activation, which is here, to the left hugely. However, they don't behave very much the same as one of the variant here, which is a V1393M, also shifted voltage dependence of inactivation to the left. At any rate, here is a summary of the rest of the variants that we can detect the currents. Overall, all but one coding changes induced at least one significant change in one of the 5 key biophysical parameters that we interrogated compared with the wild type reference channel. So half of the channel, disease-associated channel, did not express. And then for the rest of them, we characterized the biophysical properties. And then I wanted to draw your attention to right here. This is the half point of activation. So you can see with the remaining of the variants, nearly half, if not more, of the remaining of the variants shifted the voltage dependence of activation to the left. And some of them shifted as much as 20 millivolts. This is like astronomically huge for electrophysiology. So this is what we get. And we're trying to understand where in the structural domain that may contribute to different deficits in the biophysical properties. So for example, we draw these radar plots where the pentagon, each vertex represent a property. So the wild type is right at the 0 line. And then you can see the nomad, most of the nomad fall right on the 0 axis, and suggesting that across all 5 property domains, they're behaving more or less similar to the wild type channels, wild type reference channels. And then if you look at the different domains, so for example, in domain 3, a lot of variants go hyperpolarized direction. So in this case, it's basically allowing more calcium influx. And then compared to the domain 4, where there is only one pentagon plotted here, because most of the variants in domain 4 did not express or with undetectable current density that we can measure. So I'll skip this slide, and we start to get some structural insights and structural hotspot, and of where, when you have a change, then you start to modify which aspect of the biophysical property. But I wanted to share with you how do we use this data set to compare with alpha missense prediction. So this is done by Siwei in Mark's lab. Basically, we wanted to test our functional annotation. How does that compare with alpha missense? So alpha missense get 39 of the 41 impactful variants as correctly annotated as pathogenic, and then they get 1 out of 7 nomad wrong on the other direction. So it's fairly good. It's not perfect. It's okay. And one of the things I wanted to say about the alpha missense, which is depicted here, is that the alpha sense, despite its relative accuracy in predicting the pathogenicity, it does not tell you the directionality. And as I will show you later, both gain of function and loss of function are implicated in this disease cohort. And then so segregating them becomes very, very important. So after validating the alpha missense and using our functional data, we use the alpha sense to look at additional disease cohorts. So the results is here. If we use the alpha missense pathogenicity score, you can see that the CACNA1A for the missense variants is significantly enriched in the epileptic encephalopathy. And it's not so much in the genetic generalized epilepsy. Rather, the PTVs are slightly more enriched in the GGE. So right here, it seems to suggest that there is some segregation between the PTVs and the missense changes. So I'll skip this slide, but I wanted to tell you some, because we categorize different domains of the property. And sometimes so for example, if you have reduced current density, however, you still left shift in the activation threshold. So we wanted to have a method to have a combined physiological output as a prediction of how does it impact the neuronal excitability. I won't go through this, but we did that. And the reason we did that is that we wanted to be able to have a better look between the cellular phenotype from these variants, the impact of the biophysical impact with the clinical manifestation. So to do that, so here is a 3-parameter measured, and here are the 4 parameters that we modeled using this neuron simulations. And then I'll just go directly to some of the insights here. So for example, we found that current density increase and voltage dependence of activation shift is really significantly associated with a severe form of epilepsy. And then the reduction of the current density is more associated with episodic ataxia, not necessarily ataxia, but episodic ataxia. And hemiplegic migraine is really only associated, at least in this data set, which is 80 patients with 22 unique variants, is mainly associated with voltage dependence of activation. And I'll show you one example of how we start to obtain more insights onto this variant property to clinical relationship. So what I'm showing you here are 2 different types of perhaps most frequent recurring variants. Here is a A713. Here is 2 very close mutations that has very similar property, which is shown here. All of them shift the voltage dependence of activation to the left. However, the difference between these 2 classes is that the 1349Q also shift the voltage dependence of inactivation. So as the result, you can see clinically they have slightly different manifestations. So for example, A713, people who harbor A713T mutation are you see more of them with this very severe form of epilepsy, but you almost don't see them in the hemiplegic migraine. On the opposite, the R1340Q and R1352Q have a dominantly expressed manifestation clinically in hemiplegic migraine. So we wanted to do this correlation to give us a better handle on the mutation impact and the clinical manifestation. So here is what I've told you so far. We characterize a function of 50 variants. 42 of them are de novo changes of CACNA1A, whereas stricken by how impactful of nearly every one of these de novo mutations. And then we started to infer the genome type, cellular phenotype, and the human phenotype in the CACNA1A spectrum disorder, and hope to delineate a little better correlation for the physicians as well as for understanding the disease mechanism. And I would argue that understanding the function of rare coding changes of this class of protein may start to bridge the gap between the monogenic disorder and complex channelopathy. So where do we go from here? And we're grateful, very grateful to the support for the pilot from the Letter to Cure Accelerator program. So there are two key directions that we're gonna go, and we already started in both directions. So one is to identify the nodal mechanism in disease associated with CACNA1A variants and spectrum disorders. We already identified the two key phenotype, molecular phenotype. One is a voltage dependence of activation. The other one is basically we do not see much currents on the surface. So we're going after both of them, and we're starting to constructing the prototype cell lines that showing these the prototype representing the whole spectrum. And then the goal is to screen for modulators to address these two potential modal mechanisms for the CACNA1A spectrum mutation disorder. And then the second study is to collaborate with Chang Lab, as Wendy collects detailed health information and medical data from individuals with CACNA1A related disorders for the natural history study. She already collected 156 individuals in her database, and we're starting to address all of the missenses in her database. In this study we already performed, we have 20 of them, and then so we're looking to characterize the rest of them and then to map back with the clinical details and potentially identify the biomarkers. And just very quickly, here we already constructed in almost a dozen of the cell lines representing the prototype of the two potential nodal mechanisms. And we have constructed a cell line that we can read out the calcium influx through these channels using a GCAMP6 that tethered to the membrane to increase the signal-to-noise ratio. And we're very excited that this will launch us to the next stage of screen for the repurposed library and identify potentially clinical-ready compounds that we can test. So with that, I wanted to thank the 4 people who contributed majorly to this project and collaborators, Wanchi in the audience, and then Wendy and many of the support over the past few years. Thank you.
All right, next up we have Dr. Lee Rubin, who is principal faculty at the Harvard Stem Cell Institute, where his lab focuses on mechanisms of neurodegenerative diseases and identifying pre-IND compounds for translational purposes, which he has done successfully several times over the course of his career. In addition, he's an expert in modeling neurodegenerative diseases, specifically from patient-specific differentiated neurons, with a focus on Parkinson's disease, SMA, and ALS. And he's going to tell you more about his L2C project. Thank you, Lee.
Thanks, Jillian. So I'm really happy to be here today, and I'm going to talk about a project that, with the good graces of Anna and Jillian and the L2C program, we've been able to resurrect in the lab. And I'm going to, in particular, talk about a disease that probably all of you have heard of, spinal muscular atrophy. This was not so much the case 10-ish years ago, but it certainly is now. Spinal muscular atrophy and cystic fibrosis are probably the poster children for studies of rare diseases. They're not so rare in the sense of what we're talking about here today. There are about 20,000 or so kids with both of them, I think, cystic fibrosis and SMA. But it's a fairly well understood disease, and probably all of you know what I'll just quickly say right now. The disease, spinal muscular atrophy, was and is referred to as a monogenic motor neuron pediatric disease. And it's due to a reduction in the level of a protein of slightly incompletely understood function called survival of motor neuron. Survival of motor neuron is coded for by two genes, one of which is SMN1 and the other of which is a duplicated gene with a missense mutation called SMN2. And it exists in various degrees of severity, very, very, very severe, which is called type 1, until recently. Kids with type 1 SMA died before the age of 1. Then type 2, where kids would be more severely handicapped. And finally, the mildest type, type 3, where kids could live on a normal lifespan. Maybe they'd be in a wheelchair later in life. And what distinguishes 1 from 2 from 3 is the number of copies of the SMN2 gene. So this is fairly well understood. And it led to, with incomplete understanding of the disease, the hypothesis, which is seemingly very true, that the more SMN protein you have, the better you are. And based on that hypothesis, as probably many of you know, there are currently 3 approved therapeutics, one of which is a gene therapy that delivers the wild-type SMN1 gene, the other 2 of which are splicing modulators. One is an ASO, antisense oligonucleotide delivered intrathecally, and the other is a small molecule splicing modulator, in fact, a relative of branaplam that we just heard from recently, which was originally developed by Novartis as an SMA drug. Now, given what I just said, you may be wondering why I'm talking to you today about SMA. It's cured, sort of cured. And I'm going to tell you why our stubbornness about the value, the things that we've discovered using an approach that was somewhat different from everything else that was used in the field, which Jillian already mentioned, using iPS cells from patients that had different degrees of SMA severity, how that led to what we felt were very interesting insights, which is what I'm going to describe to you today. So I'm going to talk a little bit specifically about SMA. I'm going to talk in some detail about SMA motor neurons and how they behave. Then I'm going to talk about other surprising observations that we made using iPS cells made from the kids with SMA, and a general principle that I'd like to put forward today about broadening the way we think about using iPS cells. I'd also like to bring in this idea that in the case of SMA, and this is, I think, will turn out to be key to this program, we hypothesize that the carriers of SMA, the parents, of which there are 7 million in the US, are also, many of them, phenotypically abnormal and might themselves benefit from an SMA drug. So it's a little bit specific, but we're trying to establish some principles here. Okay, so what we found very early on as having made iPS cells from a bunch of kids with SMA, as I said, and making motor neurons, it was certainly the case that we could do what everybody was trying to do at the time with any disease, which is to say, you try to reproduce in culture the thing that you know about the disease. And since it was called a motor neuron disease, we tried to reproduce motor neuron death starting from patient cells. Well, this was relatively easy in SMA, unlike all the late onset diseases like ALS. ALS is another motor neuron disease, but it's genetically very heterogeneous and late onset. This is early onset monogenic. Seemed like it would be easy, and in fact, relatively speaking, it was easy. So we could show this from patient-derived motor neurons, and even here, I'm just showing you, these are just wild-type motor neurons in which we just reduce the level of SMN, and you can see that this kind of rapidly induces the death of motor neurons. Nothing at all surprising there. The thing that was surprising, though, is that we looked at the cultures in great detail by imaging, and I'm going to show you more imaging later on, now done here. What we observed was, unlike what we imagined that every motor neuron in the culture would have the same level of SMN protein, this was not true. In fact, there was wide variation in the level of protein in individual motor neurons, such that some had much lower levels of SMN than others. It turned out that the effect on motor neuron survival of reducing SMN or when studying patient cells was not uniform. And in fact, what we observed was so we plotted out the individual cell levels of SMN. So this is intensity. And every culture, whether it's from SMA iPS cells or wild-type iPS cells, had a distribution of SMN. So as a population, you could see that SMN was really low in the population, but even that severely affected population still had motor neurons that had relatively high normal levels of SMN. And it turned out that the cells that had low levels of SMN in one single well had higher probability of dying. And this was seen by following the cells over time and ultimately staining them with cleaved caspase antibody. So the effect of reducing SMN is preferentially on cells that happen to have low levels of SMN protein. Okay, that's SMA. Now, we also observed the converse. If we took SMA motor neurons and overexpressed SMN using a viral delivery in those cells, the SMN histogram would shift to the higher values, and again, that was associated with increased survival. So increasing SMN, decreasing reduced survival, preferentially by affecting low SMN motor neurons. If you shift the profile so that more cells have normal or high levels, the survival of those cells increases. In other words, in SMA, at the single cell level, the amount of SMN that they have dictates their life or death. So you can see this as an average, but more importantly, you could see this at individual cells. So why is that interesting? So it turns out every type of motor neuron culture, no matter where iPS cells came from, ALS, any genetic version of ALS, SMA, any type of SMA patient, wild-type motor neurons, whoever the donor is, we always see a histogram of levels. It's always the case that the cells in the culture with lower SMN die preferentially. And it's always the case, importantly, that increasing SMN in these cultures, even if they're wild-type motor neurons, even if they're ALS-derived motor neurons, the survival of these cells is always improved. In other words, as a summary statement, the protein survival of motor neuron does exactly what the name suggests. It improves the survival of motor neurons, regardless of where the source of the cells. Okay, so it is even true, and in fact, I won't have time to tell you about this. It is even true if you take a wild-type culture of motor neurons and treat it with toxic compounds. Toxic means that it decreases the number of motor neurons in the well. You often find compounds that decrease the total number of motor neurons while increasing the average value of SMN in the well. And the way that happens is even toxic compounds, many of which are shown here, shift the profile by selectively eliminating the cells with low SMN, leaving behind a smaller number of cells, but cells with higher SMN. So the number goes down, the average SMN goes up. So under any circumstance that we have tried, whether it's straight toxic compounds, a disease phenotype of any kind, if we make motor neurons, the higher SMN they have, the better they survive. Okay. So this I won't talk about now, but it leads you to wonder, and this is part of the new program, if that is true, it suggests the idea that a compound that increases SMN might be therapeutic for non-SMN motor neuron diseases, let's just say ALS. And if you were thinking about that, you would also have to think about what is the best way of increasing SMN in those cultures. And we, in our hands with iPS cells, the best way of doing it, even compared to the approved splicing modulator, is by blocking the degradation of SMN protein. That's part of our new project, and I'll talk about it later, but I won't show these data. Okay, so I think this was a relatively simple story, but I've summarized it here. There's cell-to-cell variation in the amount of SMN protein in every single culture we've ever seen: mouse, human, ALS, SMA. Higher levels of SMN protein increase the survival of motor neurons, full stop. This suggests that therapeutics that increase SMN may also be able to treat ALS. So that's our hypothesis. And again, what is the best type of SMN elevating therapeutic that we believe would work outside of the disease SMA? Okay. Now, there's a second part to this, and this is kind of this question: are motor neurons really the only cells affected in kids with SMA? Now, even though it's always been thought to be a motor neuron disease, there's a reason to wonder if that's really true. And one of the main reasons is the gene is ubiquitously expressed, and this is from the Broad GTEx portal. And in fact, motor neurons do not have by far the highest level of SMN. There are other types of cells that have much, much, much higher levels of SMN. So it suggested the possibility that there might be, for instance, if kids were to live longer, it might be the possibility that one would observe other tissues being affected. So our interest in this stemmed from our observation that when we made a bunch of iPS cells from kids with SMA, they were pluripotent in using the technical definition of pluripotency, which is the iPS cells made endoderm, mesoderm, and ectoderm, but they didn't do it normally. They had clear differentiation defects, and in fact, they're surprising defects. One of them is they make neural ectodermal cells, including motor neurons, much better than wild-type cells. So you generate a lot of motor neurons, they die quickly. So it's a degenerative phenotype for motor neurons, but they're born perfectly nicely. On the other hand, cells of endodermal origin are not produced well from the very same iPS cells. So just to kind of illustrate that, on the top left are endoderm cells, and you can see the SMA ones are really defective in their production. Skeletal muscle is highly defective in its production from iPS cells. I'll emphasize this now because it's very clear that kids with SMA have skeletal muscle defects independent of the motor neuron defects. And in fact, there are several clinical trials underway now to just make muscle stronger, even in kids who have received SMA therapies. So this seems to be on the right track. And we also found some cardiac defects. But the cells most affected were endodermal derived. And this shows a figure from a recent paper that we published in the Journal of Clinical Investigation with Crystal Yo. And there were two interesting things here. One is that we could reproduce hepatic, endoderm-derived liver cell defects, starting with patient iPS cells. But more importantly, it illustrates the other general finding we had, which is cells do not have equal sensitivity to loss of SMN. So in fact, the cells that are most sensitive to reduction of SMN are endoderm-derived cells. And in this particular paper, we showed a clear phenotype with carriers, heterozygous iPS cells. They clearly had a very strong liver defect, so you needed to reduce the protein level only 50% for those cells to be affected. You didn't need to go down to close to zero for the cells to be effective. So this suggests a range of sensitivity, and also suggests that if any of this iPS stuff is to be believed, it also suggests that if you look carefully at patients, perhaps you would find things that you didn't already know about. So I want to say that one again. Everybody who uses iPS cells uses them to model the parts of a disease that you understand. But for iPS cells to be really believable, you should also sort of imagine that iPS cells could tell you things you didn't already know if they're really truly representative of the patients. So that became kind of for us a big deal. Is this, it's just an artifact of iPS cell biology, it's an artifact of cell culture. And so we were quite interested in this. And finally, after a lot of struggling via Zach Ohani's lab at Harvard Medical School, with Scott Lipnick, who was a computational biologist in my lab at the time, we got access to an Aetna electronic health database with patient disease, ICD codes, patient disease diagnoses that were used for billing purposes. These records are not the best in the world, but we had 60 million of them, and from that we identified about 1,300 kids with SMA, and I'll also mention by imputation about 2,600 carriers of SMA. I'm going to come back to that. And we found, sure enough, before neuromuscular diseases were diagnosed in these kids, they had a variety of other disorders listed clearly on their health records, including cardiac and endodermal tissue-derived disorders. So that was kind of interesting. If you are imaginative enough or want to believe this enough, you could imagine that actually iPS cells, in fact, do reproduce aspects of the patient biology under the correct settings, and we can debate what those settings are. Okay, so that's second part. Multiple tissues may be affected, well, clearly are affected in SMA patients. And I want to emphasize one thing. I don't know if physicians will agree with me or not. SMA is a neurological disorder. Neurologists are examining the patients. The condition that used to be killing kids with SMA is breathing distress. I don't think the physicians are necessarily wondering about other more minor conditions that the kids have. So I want to distinguish between biological significance, things that are actually happening that are interesting maybe to some of us, versus clinical significance, the things that need to be treated immediately. Okay, and that, again, in vitro studies indicate that some cell types are actually more sensitive than motor neurons and will ultimately give rise to disease phenotypes, especially now that kids are living longer, and that there are effects that are easily observable where the cells have only lost 50% of their SMA. So this leads to this other question: are carriers of SMA really phenotypically normal? And so that's the second part of the project. Okay, so what are we doing? Part one, plans. Identify, we're interested in what regulates the level of protein, SMN protein, in motor neurons. So our plan is to separate the highest 10% from the lowest 10% and study gene expression differences in those two different populations. And then we're going to use phenotypic screens to find compounds that, let's say, derive more of the low cells into the high expressor category, and then ultimately to test compounds like that in different ALS models. And we've made a good start already, given that we had to resurrect this project from zero, even in my lab, and then transfer everything over to here. But with the wonderful help from Oyvind Gishiri and Masha Alanmo, we've already reproduced the fact that all of these cultures do have these extended histograms of SMN intensity. So that's the core of the project. So it's good that we were able to see that. We also were able to show that if you, by imaging, distinguish between the cells in the well that have the highest 10% versus the lowest 10%, and then look for, by imaging, other aspects of the cell biology of those cells, you can immediately see that there's a good correlation between, let's say, mitochondrial function, lysosomal function, and SMN. So the highest SMN expressors also have the most intact mitochondria and lysosomes, and that's kind of summarized here. And again, very reassuring that even by just straight imaging, we can already pick up differences. And Mary Rose Branch in my lab has come over here together with the people at the FACScore and has stained, affixed and permeabilized motor neurons, stained them for SMN, and then separated them by FACS. And again, we can easily separate the top, well, not easily, separate the top 10% from the bottom 10%. So that's good. So that now lets us move forward with the first part of the project. Now, part two, which is now computational, and here we're working with Ben Weisberg and Heidi Rehm from the computational group here. So we're now able, unlike when we did our first study, we can now call SMA carriers just by gene sequencing information, not the indirect way by ICD codes to get the kids and then retroactively identify the parents. So we're expecting about 9,000 SMA carriers in the UK Biobank. So that will really allow us to power up and see whether or not it's true that these parents are phenotypically abnormal in any way, using, again, ICD codes and also other informatic analyses of their health records. And then we're going to use the All of Us cohort as the validation set. And then if all of this turns out, we are ultimately interested in finding, again, a therapeutic that would treat the somewhat abnormal parents of the kids or just people who are SMA carriers. Okay, so that's it. Thank you very much. The people in my lab who did the work really were the middle three up front there. Mary Rose is in the audience, has been coming over here to do the work. Rich has been working with Mary Rose, and Chris next to her on the left is our computational person. And again, I'd like to thank the L2C program and people at the Broad that we've been working with. Thanks.
Thank you, Lee. I'd also like to invite Jen and Ricardo up for a quick Q&A.
A question to you, Lee. It'd be helpful in interpreting all of your studies to know what the protein actually does. What's the status? I mean, this has been going on for a long time.
For sure. So on the one hand, I always like to say to very good basic scientists, it shows you just don't really have to understand everything about a disease before you treat it. But on the other hand, it is really true that it's a splicing modulator, but it regulates the splicing of thousands of genes. And people have always had this idea that there must be something especially important in motor neurons, but nobody knows what that is.
I also have a question to Lee. Are you able to access information on patients, responders, and non-responders and try to correlate it with their SMN levels?
I haven't heard about a large number of non-responders to one of the treatments. It's a really good idea. We need to look at that. I presume so. A number of the patients switch therapies. And I think there are two reasons. One, they weren't working that well. And as I think the timing of treatment is so important. The longer you wait, the less that gets corrected. And the other reason is, for example, there's a supplementation of gene therapy with the oral drug. Some kids, of course, don't like getting the antisense, every 3 months by intrathecal administration. So that's the other reason for switching. But we could look into presumably how patients have responded to the treatments. We haven't done that yet. It's a good thought.
Can I ask a question as well for you? So going back to the idea of, for example, using risdiplam or branaplam,
yeah.
Are you thinking that maybe this is something that everyone should be taking a little bit, like at a low dose?
Maybe not everybody, but that's also, but it's come up every now and again. I would love to start with the more severely affected carriers, just to see how they respond. So it is true, as you're pointing out. I mean, since we've seen this on motor neurons as well, I don't know if there are situations like after traumatic damage, for example, if the nerve is cut to preserve the cell bodies better, maybe it would be useful under those kind of circumstances as well.
For a fantastic session. The speakers will hang out for a little while to answer any follow-up questions. Please be back at 1:15 so we can continue with our program. Thanks, everyone.